Imagine living in the 1980s and being told that computers would take over everything, from shopping to dating to investing in the stock market. The billions of people would be connected through some kind of network, that you would own a mobile device many times more powerful than supercomputers. It would have sounded absurd, but then all of it happened. Science fiction became our reality, and we don't even think about it. Today we are at a similar point with genetic engineering, so let's talk about it. Its origins, what we are currently doing, and a recent discovery that will forever change how we live and what we consider normal. Humans have been modifying organisms for thousands of years. Through artificial selection, we have strengthened useful traits in plants and animals. We became good at it, but we never really understood how it worked, until we discovered the code of life, deoxyribonucleic acid, DNA. A complex molecule that guides the growth, development, function, and reproduction of everything that is alive. Information is encoded in the structure of the molecule. 4 nucleotides pair up and create a code that carries instructions. Change the instructions and you change the being that carries them. As soon as DNA was discovered, people tried to play with it. In the 1960s, scientists bombarded plants with radiation to cause random mutations in the genetic code. The idea was to randomly get a useful variation of the plant. Sometimes it actually worked. In the 1970s, scientists inserted DNA fragments into bacteria, plants, and animals to study and modify them, for research, medicine, agriculture, and even just for fun. The first genetically modified animal was born in 1974, turning mice into a useful and standard research tool, saving millions of lives. By the 1980s, it became commercial. The first patent was granted to create a modified microbe that could absorb oil. Today, we produce many life-saving chemicals using genetically modified life, such as clotting factors, growth hormones, and insulin, things we once harvested from animal organs. The first lab-modified food was sold in stores in 1994. The Flava Sava tomato, which had a much longer shelf life due to an extra gene suppressing a rotting enzyme. In the 1990s, there was also a brief attempt at human engineering to treat maternal infertility, which led to babies carrying genetic information from 3 humans, creating the first humans with 3 genetic parents. Today, we have super-muscled pigs, fast-growing salmon, featherless chickens, and transparent frogs. And for fun, we've even made things glow in the dark. You can buy a fluorescent zebrafish for less than $10. This is already impressive. But until recently, gene editing was expensive, complicated, and time-consuming. That has now changed with a revolutionary new technology, CRISPR. Overnight, the cost of genetic engineering dropped by 99%. Instead of taking a year, it now takes just weeks to conduct an experiment, and basically anyone with a lab can do it. It's hard to express just how revolutionary CRISPR is. It literally has the potential to change humanity forever. So why is this revolution happening so suddenly, and how does it work? Bacteria and viruses have been fighting since the beginning of life. Viruses called bacteriophages hunt bacteria. In the ocean, they kill 40% of them every day. Viruses do this by inserting their genetic code into the bacteria, hijacking them and turning them into virus factories. The bacteria try to resist, but they almost always fail because their defenses are too weak. But sometimes a bacterium survives the attack. If it does, it can activate its most effective antivirus. It stores a piece of the virus's genetic code in its own DNA, in a file called CRISPR. There, it's stored safely until needed. When the virus attacks again, the bacterium quickly makes an RNA copy of the stored DNA and uses its secret weapon, a protein called Cas9. Cas9 scans the inside of the bacterium, searching for signs of the invader, comparing every DNA sequence it finds with the stored sample. When it finds a perfect 100% match, it activates and cuts the virus's DNA, making it useless, protecting the bacterium from the attack. What makes Cas9 special is that it's extremely precise, like a DNA surgeon. The revolution began when scientists realized that CRISPR is programmable. You can give it a copy of the DNA you want to modify and insert the system into a living cell. If old gene manipulation techniques were like using a map, CRISPR is like using a GPS. Besides being precise, cheap and easy, CRISPR offers the ability to edit living cells, turn genes on or off, and target specific DNA sequences. It also works with any type of cell, microorganisms, plants, animals, or humans. But as revolutionary as CRISPR is, it's only a first-generation tool. More precise tools are already being developed and used as we speak. In 2015, scientists cut the HIV virus out of living patient cells using CRISPR, proving that it was possible. Just a year later, they conducted a large-scale experiment on rats infected with HIV. By injecting CRISPR into the rats' tails, they were able to remove more than 50% of the virus from their cells. In a few decades, CRISPR could cure HIV and other retroviruses. Viruses that hide in human DNA, like herpes, could be eliminated this way. CRISPR could also defeat one of our worst enemies, cancer. Cancer happens when cells refuse to die and keep multiplying while hiding from the immune system. CRISPR allows us to edit our immune cells, making them better cancer hunters. Getting rid of cancer could one day mean just receiving a few injections with thousands of your own cells modified in a lab to cure you permanently. The first clinical trial for a CRISPR-based cancer treatment in humans was approved in 2016. Not even a month later, Chinese scientists announced they would start treating lung cancer patients with CRISPR-modified immune cells. Things are moving fast, and then there are genetic diseases. There are thousands of them, ranging from inconvenient to deadly. More than 3,000 diseases are caused by a single incorrect letter in your DNA. We are already developing a modified version of Cas9 that can fix that single letter and cure the disease in the cell. In a decade or two, we could permanently cure thousands of diseases. But all of these medical applications have one thing in common. They are limited to the individual and die with them unless we use them in reproductive cells or very young embryos. And CRISPR can and will probably be used for many other things, like creating genetically modified humans, designer babies, a gradual but irreversible change to the human gene pool. And this is where things get controversial.